Hello everyone. I'm Kalaivani Raju, a senior bioinformatician at Mogrify Limited, a Cambridge-based disruptive startup that aims to transform cell therapy by systematic discovery of novel cell conversions. I'm presenting a poster on Mogrify computational framework to convert between cell types, wherein I hope to outline the current practices in gene and cell therapy, introduce the Mogrify platform, its theoretical foundations and experimental validations, mention the projects in pipeline, and finally discuss how Mogrify will impact the cell therapy space. Despite major investment in the field, few cell therapies have gained regulatory approval with limited commercial success. The success of therapeutic products is defined by three factors: safety, efficacy, and scalability. Achieving these for cell therapy presents new challenges like overcoming transplant rejection, producing and culturing functional cells, and developing an off-the-shelf therapy with a scalable manufacturing process. In gene therapy, the therapeutic agents, represented here by a virus or a lipid nanoparticle, is injected into the patient. The therapeutic agent carries a genetic cargo to correct the genetic defect and may have a tropism for a specific organ, like, say, the liver. Once the genetic cargo is delivered, the genetic defect is rescued. In cell therapy, the therapeutic agents is the cell that have been genetically manipulated and expanded ex vivo so that they acquire a new therapeutic function. In autologous cell therapy, where the donor and patients are the same individual, after development in the academic or clinical setting, all the functional units are located at the hospital site nearby. This allows a close interaction between clinical, manufacturing, and analytical teams to ensure that the vein-to-vein cycle is completed in time. This model is costly due to small scale of operations. In the hybrid model, the manufacturing and analytical functions are centralized so that cells from patients collected from different hospitals are then transferred to a centralized structure equipped with manufacturing and analytical functionality. This model benefits from large-scale centralized production, but it relies on excellent transport system, chain of custody, and chain of identity for its success. The cost remains high. In the case of allogenic cell therapy, where the donor and patient are different individuals, the starting cell material, which is either from a donor or a cell line, is very suitable for a scale-up model where all the functionality for manufacturing, batch testing, and releasing, as well as storing, are in the same manufacturing center. Automation, large batch production, and a strong organized supply chain help reducing the costs. In this context, I would like to present our computational platform Mogrify, which can convert between any two cell types or cell states without having to go through the pluripotent stem cell state. Mogrify has developed a systematic big data science approach powered by next generation sequencing and gene regulatory data to identify the optimal combination of transcription factors or small molecules needed to convert any mature cell type into another mature cell type without having to go through the pluripotent stem cell or even a progenitor cell-like state. The main steps of Mogrify algorithm is depicted here, and it includes calculation of contextual differential gene expression, identification and scoring of genes which need to change, defining local regulatory network influence of each transcription factor, scoring the conversion potential of each factor in isolation, and optimization of network coverage and overlap of combination of factors. Underpinned by a decade of development through an international research effort, Mogrify is a computational approach powered by genetic, proteomic, and epigenetic data to systematically control the cellular transcriptomic network underlying cellular identity. This platform can identify key factors required to culture or convert any cell type to any other cell type. We have applied Mogrify to 173 human cell types and 134 tissues, defining an atlas of cellular reprogramming, including both known transcription factors used in transdifferentiations and new ones never implicated in cellular conversions. Mogrify in silico predictions have been validated in vitro in over 20 different cell conversions, including generation of endothelial cells, astrocytes, and cardiomyocytes. Mogrify predictions are shown here compared with previously validated conversion protocols. The factors predicted by Mogrify for each of these conversions is highlighted in orange. The factors in blue, which were not predicted, were shown to be upstream of other factors that are required for the conversion. For example, NANOG instead of KLF4 and CMYC, while the factors in red were shown to be unnecessary towards driving the conversion. So the company is deploying this platform to develop novel cell therapies addressing musculoskeletal, autoimmune, cancer immunotherapy, ocular and respiratory diseases, as well as generating and licensing novel IP-related cell conversions. This can exhibit safety, efficacy, and scalable manufacturing profiles suitable for development as cell therapies. One of the edges Mogrify has is that any cell type can be used as the stock. Large quantities of well-characterized and reproducible batches of desired cell type, these cells can be used directly for regenerative medicine, for example chondrocytes, or as a basic frame to be empowered with further functions, for example CAR T cells. If you have any questions, please feel free to email me at kalai@mogrify.co.uk. Thanks for the opportunity and time.